Welcome back to At the Bench, the physician scientist podcast of the Incubator. I'm Misty Good. I'm the division chief of neonatology at UNC Chapel Hill, and I'm excited to be co-hosting today with Dr. Betsy Crouch. Betsy, would you like to introduce yourself?
Of course. Thanks everyone for joining us. I'm Betsy Crouch, as you all know. I'm a neonatologist, a neuroscientist, and a vascular biologist at the University of California, San Francisco. I'm very pleased today that we get to interview my friend and colleague, my up in the middle of the night, taking care of moms and babies, collaborator, Dr. Stephanie Gaw. Dr. Gaw, would you please introduce yourself?
Thanks for the invitation to chat with you guys today. My name is Stephanie Gaugh. I'm an associate professor of maternal-fetal medicine at UCSF, and my lab works on placental infections and maternal fetal immunology. Um, yeah.
Yeah, thank you so much for joining us. I'm really excited to talk about all of the different agents and infections that you work on, many of which, are are coming. What's the word I'm trying to look for? Are like rising in their incidence amongst both pregnant people and the babies that we take care of. And so this is really important work, and we're excited to talk to you about it today. Um, I think why don't we get started sort of on your career trajectory? Did you always know you were going to be a physician scientist? How did you decide to be an MFM specifically?
Oh gosh, this is such a roundabout story, and I've been talking about this a lot actually because my oldest daughter is applying or is graduating from high school and went through all this stuff about choosing her major, and I have basically told her like I had no idea that the job I have existed at at your point in life. I went to Berkeley for undergrad and I majored in molecular cellular biology. I still don't know exactly why. And that's when I first had a taste of the lab. I did my senior research thesis in an immunology lab with Dr. Neelab Shastri. We studied very basic immunology, MHC, antigen processing and presentation. But somehow during college, I decided that after I was going to join the Peace Corps. And so I wasn't applying to medical school or anything. Um,
Wow.
and I hadn't really decided what I was going to do, but I was like, I just needed to get away. And I wanted to, I remember telling my friends, I wanted to experience the world and like know what like most of the way most of the world population lives. So I graduated from Berkeley. A lot of my friends, of course, were applying to medical school, and I joined the Peace Corps. I was in Honduras for 2.5 years. And it was actually there that, back then there wasn't cell phones or even email was very rudimentary. And so my good friends wrote me letters, like real letters that took a couple months to arrive. And so one of my friends had gotten into an MD PhD program, friend from high school, and so he told me a little bit about that. So that's the first time I'd heard of it. Um, but it was there I think I was deciding between medical school and doing research versus doing some sort of public health something. And I think actually somehow during my time in Honduras, I decided I wanted to do biomedical research, but on a topic that was directly applicable to the developing world. And so that's how I got into infectious diseases and global infectious diseases specifically. I applied to MD PhD programs because my friend told me about them, and he had a great time. And so I ended up at Albert Einstein College of Medicine in the Bronx, New York. And I was really drawn to that program because they were really amazing. Immunology and infectious disease and parasitology department because it's probably one of the most diverse, as in from all over the world, um, academic medical centers in the country. And so they would see all these immigrants and travelers, and people went back to visit families all over. So they see all the weird stuff. And there I joined a malaria lab, and Dr. David Fiddock, who's now at Columbia, and I studied drug resistance. It's very molecular parasitology. I made basically transgenic parasites that had different mutations for drug resistance genes to see their responses. Very in the lab, but um, that kind of got me into infections and infectious diseases.
Do you
I
have any
found
crazy stories from, I don't know,
no, it
from your
just like
time
it
in?
was basically like I uh, God bless all the people that work in malaria labs, but I would like never want to grow the malaria parasite again. It's like we
It's so it's so
every
hard to grow
and
them, right?
yeah, we had to look every week. Like we were just like, if you had to go somewhere to get somebody else to take care of all your parasites, cultures, and stuff. So I've been like, I'm never growing it again. We'll like collaborate with someone that grows it.
But wait, you still do work, you to
I work
clarify,
it. I work it. I don't culture the
you
parasite.
don't culture the
Yes,
parasite. Okay.
very finicky, very
Yeah.
finicky. And you have
Can I
to,
ask a
yeah.
Can I ask you a question before you, like, go on? Because I think this is really important for everyone who's, thinking about research and trying to pick a project. How did you pick a malaria lab?
Mm.
Oh yeah, I think I rotated through. So we had to do 3 rotations, and I rotated through an HIV lab first, and that was fun. And then the malaria lab was the second one, and the third lab was a tuberculosis lab. And I think a big part of it was I just really clicked with my mentor. I think we had similar personalities, and I felt the least nervous around him, to be honest. Where I could ask him my dumb questions, he would ask me dumb questions. Like it just seemed very like relaxed and comfortable in that way. And then scientifically, I still think, the Plasmodium parasite is probably one of the most fascinating pathogens like all around. It's so complicated. The life cycle so complicated. Each life cycle has a totally different, set of genes that's expressed, and that's really why it's basically like caused so much harm to humankind over millions and millions of years, I think. And so that was really interesting. Um, and it just seemed a little different, I guess, and it was really, I was very committed to global health, I think. And that was clearly one of the diseases that like only happened in the poorest countries in the world.
Mm, awesome.
Where was I? I think so I did my PhD. Oh, and then I thought I was going to do medicine and infectious diseases and do that more traditional physician scientist track. Um, but I defended. My first rotation back was OB-GYN, and I loved it. But I thought it was because it was like I was out of the lab finally. And finally, like my first rotation. I'd had a baby at that point. I had a 1-year-old, and so I was like, oh, I'm just comfortable with like, I've had a baby before, so I'm not totally clueless. And and so I really enjoyed it. Then I did something else and then medicine, and I just couldn't do it. I was just really cranky all the time. I think clinically, I did not enjoy clinical medicine very much. So I had this crisis, and then I thought, oh, I'll do pediatrics and do peds ID. And then I had my peds rotation, and I also could not take care of kids. It's just one of those, you just find your patients. And I got really good advice from one of, how you collect like little tidbits of advice from, people that you sometimes encounter just once, and sometimes like they're your more long-term mentors. But it was somebody who came to visit as a visiting speaker to the MD PhD program. And she said, um, she said basically you should pick your clinical specialty by the patients you want to take care of, not by the research you want to do, because um, the research questions will come and research can take you anywhere, right? You don't know where, the science is going to take you, but you can't change the patients that you take care of. And also, if your heart is in research, if you don't enjoy the patient care, you'll feel resentful of the patients in the clinical care that you do kind of in conjunction, side by side, or um, if you're doing both. And so, take care, pick patients that you enjoy taking care of, and you won't be like hate half of your life or whatever percentage of your time that is. Anyway, I thought that was really great advice. And so when I couldn't do peds and I couldn't do medicine, I was like, oh, maybe I'll do OB. And I went and talked to my program director, and I was like, I'm so sorry. I think I'm going to do OB. And he was like, that's great. And, I think that like that week in, I forget, Science or Nature, they had published like the angiotensin 2 receptor as like a potential mediator of preeclampsia. I was like, look, there's science. And then the more I thought about it and got into it, it just made a lot of sense. And maternal fetal medicine especially attracted me because it's basically all the fun parts of medicine and peds kind of all in one patient. So all the medical complications and like the scientific and clinical questions just get like 10 times more interesting because you're like balancing two different two different people at the same time. And of course, everything that a mom gets or a person gets that's, a woman gets that's not pregnant, when they're pregnant, they may also have that complication. There's definitely diseases that specifically target pregnant people. And then, of course, the ones that, have very little impact on the mom, but definitely impact the baby. So it's kind of a really nice space. And then I think clinically, we talked a little, it's just the pace just made a lot more sense to me. I think I'm very impatient. So, like a short period of time taking care of those patients really fit me. It wasn't this like more medicine. A lot of it was the chronic illness I did not do well with.
There
Can
is
you
so much. Yeah, I know this is such a rich
Mhm.
area, I think,
Yeah.
to really
Mhm.
dig into, and it's
Good.
so enjoyable to have an MFM also, I think, on this podcast because you might be our first MFM, right, Misty? Am I correct?
Yes. Yes, I
Um,
believe so, yes. It's
Honored.
exciting.
yeah, but, it's a podcast about neonatal physician scientists or actually physician scientists who also work in adjacent areas to neonatology, and I think one of the reasons why we're friends is because like, I love to pick your brain about the way that you're thinking about, I mean, both medical and scientific problems. So, and I don't know, I just was curious about you reflecting a little bit. I do feel like physician scientists are a rare breed in MFM, although there are outstanding programs like the RSDP, excuse me. Um, and I wondered if you could talk a little bit about that training program because you were part of
Yeah.
that training program and, other efforts to cultivate more physician scientists and
No,
maternal fetal
definitely. I
medicine.
think there's very few physician scientists in OB in general and definitely in MFM. So it's hard to find role models. And and it's hard to kind of like maintain a job or have a job because, at every institution there's just I mean, here there's just one of me. And so I think like my, division director, my leadership doesn't know exactly how to take care of me, and there isn't like a direct mentor within the department that physician scientist wise that has gone my career track. I think the RSDP, the reproductive scientist development program that Betsy brought up is it's a national K-12. I think it's similar to the PSDP, the
It is,
one, right?
yeah. In
Uh,
fact, the reason I know about it is because I was a part of the PSDP, and, I think they were trying to collaborate with the RSDP, which makes a lot of sense,
yeah.
and they invited me to speak at the RSDP, um a retreat, which was during COVID, so it was online. So I have all this RSDP swag, which I really appreciate. And it makes me think fondly of the MFM folks because as a neonatologist, right, in the spectrum of pediatrics, the neonatologist is the closest to the the OB space. So anyway, but I really enjoyed it because of course, as neonatologists, we do work closely with our OB colleagues.
No, so, that program is a national K-12, NIH funded, but they also get supplemental funding from Bros Welcome Fund, from March of Dimes, to kind of, I think, to broaden, and amplify their resources. And it's all OB-GYN specialties and everybody's a physician scientist, essentially. There is a lot of MFM representation, I think. And I think, programs like that are really important, especially for specialties where there are few physician scientists. 'Cause I will say that's how I know like almost all the physician scientists in MFM in the country, because almost all of them had either I met during the interviews for RSDP or like through the program over the years. So it's really great to have that network across the country as we kind of we're all applying for the similar career development awards and getting all the rejections from your first grants and like getting your grants and starting your lab and kind of it's nice to have a sounding board for that, especially being one of the very few in my department, even at a big research institution like UCSF.
I think for the RSDP and also the PSDP, like, it really talks, like, and gets people, I think, aware of the importance of peer mentorship, especially if you're the only person at your institution, as you were saying, who's a physician scientist or not necessarily your institution, but in your division. I think it's nice to be able to network with people who are going through the exact same thing or have just gone through it, those near peer mentors. And so I was just hoping you could talk a little bit about that in terms of like, when you're an early career um, physician scientist, how to take advantage of that networking opportunity or those early collaborations.
Yeah, I mean, it was really instrumental for me because, as an MFA, like UCSF, amazing, research institution, biomedical research. I'm the only MFM physician scientist here. We do have other ones in different specialties, but, all the different subspecialties are very different in their clinical practice, what you need to like maintain skills versus, how to balance clinic and all that. So I think a lot of, I think my first 2 years as faculty here, I didn't really know what I was doing yet. And my division also didn't know what I was doing. So, like, for example, you're on a K. I was on, RSVP at 25% research time or clinical time, right? I was scheduled exactly 25%, but I'd have like a clinic here, a clinic there. And like in MFM, we do a lot of satellites. So I'm like driving around town. And I was like, this doesn't, and I think part of it's just talking to my friends at other places, like, what do you do? And it finally came down to like, everybody does something different at each institution, but kind of understanding just kind of the flow of, my day to day. So, now I block my time. So I do 4 days of clinical a week, or sorry, a month at, and I have basically all of it blocked that time. And and so I kind of feel like I just disappear for 4 days a month, and then I'm back all the time. Other people will do like one specific day a month, or sorry, a week, and like every Monday, they're in clinic. Um, and I think a lot of that, talking through it kind of depends on the what kind of research you do in the lab and how your experiments are set up, right? Whether you need the continuous days, but it was definitely the breaking up of everything. Well, like you're doing notes like the next day because it's just driving around town, was definitely a time suck. So, so it's good to, I think it's always good no matter what specialty you're in to talk to other people about, how they structure their day, how they manage their time, and what resources they have, and maybe different ways to, because those little things, like, my division director never had a handbook, like, this is the what, if they're in the lab, they should have, they didn't think to ask me, and I didn't, it was my first job, so it's like, I'll just do whatever you want me to do. And it took a while to figure out, what would work.
Well, when you're driving around to all those different places and obviously doing your clinical work, we're curious about any clinical observations that you made that have motivated your research and the work that you're doing now.
Yeah, I think I think one of the funnest parts of my job is that, well, in OB-GYN, especially with them, I feel like we're like 20 years behind pizza medicine in evidence-based medicine. There's a lot of dogma. There's a lot of just this is the way we've done, and we're scared to change clinical practice, right? And so there's a lot of unanswered questions when you step back and think about, why we do things a certain way or the explanations we give patients for, why this happened or that happened. And there's so many unknowns. And I think that's where, I feel like my secret power as a physician scientist really comes in because you put your like scientist hat on, you're like, oh, how could we answer that question? Could I do it in the lab somehow? What samples do I need to collect? And that's been really fun, and I don't think when you're starting out early, you really understand that, because I feel like you're comparing yourself to, these amazing clinical mentors that you have that, they know everything about every single condition, and then, like, these amazing scientists that know, like, all the latest technology, and I'm kind of, like, in the middle where I don't really know everything about anything, but I really can translate right between, the bench and clinical medicine. Recently, we started working on um, sepsis in pregnancy, just infections in pregnancy, or even just the fever in pregnancy and the inflammatory response. And there's been a lot of, I don't know, kind of epidemiologic studies showing that, obviously sepsis in pregnancy is bad neonatal outcomes. Most of that work has been in with women that have sepsis, and then they're so sick they deliver at that same time that they are actively septic. So of course the babies are really sick, very often preterm. And I remember I was on service where to clear this patient who was admitted was, septic in the ICU for like 2 or 3 days with pneumonia. And then she was getting discharged. We're like, "Oh, great. Congratulate, you're going home. Everything's back to normal. We'll see you in the clinic." And she's like, "Is everything going to be okay with my pregnancy?" I was like, "Oh, yeah, you're going to be fine." We walked out of the room. I'm like, "I think we just lied to her." I was like, "I don't think we know, actually," and then we looked it up, and there'd been no studies on antepartum sepsis that resolves prior to delivery, like, nothing at all, and so that started me like, "Oh, let's look at, like, some databases," look at what epidemiology databases, and then that's when I started to collect samples, and so, of patients that had some sort of septic event during pregnancy, collected some of their blood samples then, and then their samples at delivery. And, with some of the database studies, we looked with the California Birth Registry and patients, that had sepsis at the time of delivery. There are increased risk of all these neonatal outcomes that are, after controlling for gestational age, even retinopathy of prematurity was like weird. It's kind of weird, like what happens when we looked at the UCSF database over, I think, 20 something years and looked at patients that were admitted for sepsis, discharged, and delivered later. There's a higher incidence of growth restriction, hypertensive disorder of pregnancy, just something more chronic. And so, right now I have a fellow in the lab who has been collecting non-transmitted infections. So basically pyelonephritis or a kidney infection during pregnancy where, the babies don't get the E. coli kidney infection, right? But the moms can get really sick. And we're, doing spatial transcriptomics on the placentas right now to see what happens with that, that inflammatory insult in the middle of pregnancy to impact how the placenta continues to develop over the rest of gestation and impact pregnancy outcome.
Yeah, what cool stories. I will say that that's one of the ways also that we know each other is that Stephanie and I share a spatial transcriptomics machine, and it's been really great because I'm interested mainly in vascular cells in the brain or the CNS, and Steph's really interested in them in the placenta. But, I think, we have 2 postdocs who are working together now, and it's really useful to compare vascular phenotypes in different states, in different organs, and I think it's just another story about how you have maybe a clinical commonality that leads to, like, research discovery too. Um, yeah, go ahead, Steph.
Yeah, I was gonna say, and it's also, when you're learning a new technology and nobody really knows what they're doing, I feel like it's
Yes.
nice to work through it with somebody else that you trust and
Yes, exactly.
and think through things together.
It yeah, yeah, I mean, there's been so much interesting work, I think, in the spatial transcriptomics field and, like, moving from I'll just say this, this, a topic for a different forum called spatial transcriptomics, which is not, um, we need to move from, just looking at dots, which capture the cell that's the majority in that tissue, to really a much more targeted approach that people are doing in different ways, but yeah, I found that to be very exciting.
I do think it's important though to know, like, from an immunologic perspective, like what is there, right? And for the placenta in particular, there's so much unknown about like not we know what it does functionally, but in the setting of infection or sepsis, maternal sepsis, as you mentioned, what do we not understand about what immune cells are there? What are they doing? I think the placental response to infection and how that affects the baby is critically important.
Yeah, and I think, a lot of the work, I fundamentally, the problem we always have with placental like, translational studies in human placentas is that you don't get the placenta till the end, right? And you can't tell what happened early, and if you do, have a miscarriage or an abortion early, that you don't actually the abortion samples, you don't know what their outcomes would have been, right? So it's always trying to make some sort of inference from the endpoint. Um and, I don't have an amazing answer for how to overcome it, but just keeping that in mind, I think that, we're doing we've recently published this epigenetics paper looking at, so placental malaria is the leading cause of fetal growth restriction worldwide, right? And then, of course, we have fetal growth restriction in the United States that's not malaria related. And, you know, epigenetic modifications in the placenta have been associated with fetal growth restriction. Um, in studies in developed countries, but no one had looked at it in malaria. So we wanted to look at, fetal growth resistance with and without malaria. So like a cohort of samples collected from here, in San Francisco, and a collection of samples that we collected in Uganda that had malaria. And basically, the epigenetic the differentially regulated genes, CPG sites were totally different in both cohorts, even accounting for site, just environmental, like different populations. And so, that's really interesting. Kind of we're trying to build on that now and think about why, but that the mechanisms of fetal growth restriction here, in non-malarial versus malarial growth restriction is very different for some reason, even though it's kind of the same end phenotype. Um, but you can't say what caused it, right? I think this is what, when we're writing the paper, like, we don't know what came first. Like, are the epigenetic changes because of growth restriction and the placenta is like compensating for it? Or did those happen for some reason, like because of malaria and then lead to growth restriction? Like, we can never really say, we can't establish causality with that one
But you
sample.
can in an experimental model, getting back to your point, right?
Yeah, so I think I you know, I would joke because I spent probably the first 3 to 4 years of my faculty career just collecting a bunch of samples that are interesting and not really knowing what I'm gonna do with them. But I think the next one that I'd like to start is doing a bank samples. We do have some patients that get chorionic villus sampling, CVS sampling early in pregnancy, and just starting to bank those because if they get something or not, at least we have like a beginning and an end point in the same pregnancy where we can maybe try to find predictive changes over gestation. But it's kind of one of the more challenging and but also kind of intellectually um, interesting parts of working with placental research. Right, we're getting this one time point, and we don't know what happened first.
Can you talk a little bit more about your biorepository that because you're collecting samples, but the
Yeah.
biorepository that you've developed sounds interesting, and certainly for our listeners, you said the first 3 years of your faculty position, you were just collecting samples, and I'm wondering, are you doing that now in a more focused way or a broader way to be able to, like, you never really know where your science is going to take you, and so just thinking about the next study or, the next grant, for example, like, how do you decide what to collect and when to collect at different time points?
And this is Steph's superpower. What a valuable resource, right? And the fact that she had the vision to start to bank these samples with now like this, placental biobank with how
We
many
have like
cases?
thousands. Well,
Yeah.
so I think when I first got into, so I was a malaria researcher. We like cultured parasites. Very lab based, and then in OB, and when I came to residency at UCSF, I worked with Susan Fisher, who's a placenta biologist, and just out of luck, honestly, she had a graduate student that was interested in placental malaria and had collected some samples from the Democratic Republic of the Congo. They all had symptomatic malaria at the time of delivery, but almost no clinical data. So, we did some, like immunohistochemistry staining for like macrophage markers and kind of very beginning things. But that was when I was like, I can't do any like molecular testing on these samples. Like, at that time, spatial, there wasn't any technology for FFPE bank samples. And also, there's almost no clinical data. So we just knew the gestational age that they say they were at and that they had malaria. And so that's when I started thinking about collecting samples that would be really useful in translational research. Because I do think this is one of the major problems when you go back and look at all the translational work that has been done, in the perinatal medicine field. Like, for example, preterm birth is like classic. A lot of places people will define preterm birth as if you delivered before 37 weeks. But of course, there's a huge difference between spontaneous preterm birth and then preterm birth for some other reason, right? And, they're just kind of grouped together, we don't know, or like labor or non-labor, right? Like, especially when you're studying inflammation and immunity, like the labor process itself triggers a lot of cytokines and, cortisol and all this inflammatory response that you wouldn't necessarily see in a non-labored, patient. And so I think that's when I started to set out and think about how do we want to preserve all these samples to potentially use for all these fancy experiments that we want to do down the line, whether it's like, advanced immunology experiments, transcriptomics experiments, either in blood samples or in the placenta, and then obviously imaging and and all this technology that when I was started collecting, I had no idea we'd be able to do. And so I have collaborators that do run clinical trials in Uganda malaria, so that's how I was collecting a lot of the malaria samples. But um, especially unknowingly, it's always nice to have some sort of domestic kind of connection, So again, because malaria is the leading cause of growth restriction, I'm like, oh, I just started collecting growth restricted babies here of all types, and it's like every once in a while. And my selection criteria at the beginning was because it's just me and I had one technician maybe at the time was like, well, whenever they were available, we just like, oh yeah, let's grab that sample, right? And over time, and I think really actually when my biobank really grew was during COVID. We got a pot of money. At the beginning of the COVID pandemic, it seems so long ago now, but people were really motivated, I think, to do research and do research quickly. And I've gained some experience from the Zika pandemic of kind of collecting, exposed samples really quickly and enrolling a cohort really quickly. So we started enrolling these COVID patients. So at the beginning, it was just everybody with COVID, that tested positive for COVID. We collected blood samples at the time of infection. We like isolated plasma, PBMCs, like just to be able to do anything. We collected swabs, we collected meconium, stool, like this everything. 'Cause we didn't know what we were gonna do with them, but I was afraid to not have something, right? I was thinking about like, what if we want to grow it out? And what if it like, the report came out that it was, people in the ICU were COVID positive in their stool. So I'm like, oh, maybe it's transmitted that way. Let's collect, meconium and maternal stool and we'll see. And we collected everything. And then probably about when the vaccine came out, we started doing that too. Like, oh, maybe we should look at vaccine responses. Because actually, it was a beautiful natural experiment, right? Of like the first antigenic sin in humans of this brand new pathogen. And we knew exactly when during pregnancy they were exposed, right? For initially the infection and then with vaccination. Then when the vaccine came out, we realized, oh my gosh, there's going to be no more COVID negative patient, like naive patients. So then we called them unicorns. So then we were like selectively looking for people that said they hadn't gotten COVID and were not vaccinated and like begging them for samples. And now those are like our most precious samples because otherwise everybody else is like COVID serology positive and you don't know how many times they've been exposed. And so, over the past 5 years, you know, that also expanded when the RSV vaccine came out, where we wanted to, look at RSV. And again, that's a different um, scenario in that everybody has baseline immunity to RSV, right? So you can't look at naive responses, but it really is kind of booster responses, uh, during pregnancy. And I think we spent probably 2 years collecting samples. We didn't have the bandwidth to do anything else, and then really started digging into a little bit about, looking at immune responses, and the most economical and fastest thing to do is to look at antibody responses first. So, we looked at, vaccination versus infection and also over the course of gestational age. And I think these are kind of the closest thing we'll have to infecting pregnant people, at a specific time with a novel pathogen, right? To look at that pathogen specific responses and seeing that, what we found, I think is really interesting is vaccine induced antibody responses are always higher than natural immunity for some reason. Neutralizing activity is higher in vaccinated responses, and antibody transfer is also better with vaccine induced antibodies than naturally induced antibodies, which I think is really interesting. We don't know why. Um, but as far as for vaccine development in the future, as we look at how more vaccines can benefit not just the mom, but really their baby.
Yeah, the baby.
Yeah. As a clinical point, like a lot of moms won't vaccinate for themselves, but they will for their baby. Um,
Mhm. Do you have speculations, hypotheses about why the vaccine-induced, antibodies are more easily transferred and have an increased antigenic response?
I I think I don't know exactly why. I think we're looking, my main R1 right now is looking at placental factors that dictate differential antibody transfer, whether it's different. We also, when we look at epitope specific transfer, there's also a difference in the epitopes that are transferred from mom to baby and a difference for every patient. And so we're trying to like dig into it a little bit more, whether it's the antibody subtypes that are generated or something else that we don't know anything about, right? A lot of maternal fetal immunity is kind of hand wavy, like the antibodies go to the mom or from the mom to the baby and the babies are protected. We do know that, but how it happens, we don't really know. And and I think, I suspect it has something to do with the gestational age at the time of exposure. And I do think there's something about, when you're getting a vaccine, you're otherwise healthy and kind of like your body generating antibodies under duress, like with an actual infection, the quality or like the repertoire might be a little bit different. But I don't have anything more intelligent to say about that. That's like the only thing, right? Like you're totally healthy, you get exposed to a really high dose, and it might be a dose response too, right? A higher inoculum of the vaccine antigen.
Well, I think those are brilliant insights, so thanks for sharing. I guess the only other infection that I'm aware of that you study that we haven't talked about is syphilis, which I think, is a rising public health problem. So I also wanted to take the opportunity to pick your brain on, your efforts,
Oh, for
for
syphilis. Well,
yeah.
syphilis I've been fascinated by for a really long time because, 1, it's such an old infection. 2,
Mhm. Yeah,
it's
it is.
mostly poor people that get it. And 3, and it's like the great imitator, right? It, like, infects everything. It's so bad for babies. It gets in the placenta, and, like, we don't know what happens. And nobody studied it in millions or not millions, but many, many years, right?
Yeah.
And
Also very hard to culture.
right, they, like, just learned how to culture it a few years ago, actually, and there's just a handful of labs that can culture it. I think, we have a grant right now in R21 to collect, syphilis samples, and this is more of a clinical translational study because, diagnostically, it's impossible to diagnose, recurrent syphilis in pregnancy, and, like, the consequences are so severe because if you follow the guidelines, you're like, "Oh, if it's, like, early, then you do 3 doses. If it's late, just do 1 dose." But you can't really tell because it's based on RPR, which is a totally nonspecific antibody, and then also even with the specific antibody, it's like it makes no sense, right? So I was like, "Why don't we have a better molecular test or something else?" So, I work in Fresno in the Central Valley, which was or is one of the syphilis hotspots in California. And so at the hospital that I work at, community regional, um, Fresno County, I think had the second highest number of congenital syphilis cases in 2020. The last time I looked, this is when I wrote my grants. There were like 240 congenital syphilis cases reported to the county. So you can imagine the maternal syphilis cases was more than that. And of those, 90% of them delivered at this hospital that I work at. So, the numbers have thankfully gone down significantly because their public health department did a lot of really good work for outreach and education.
Go
But
public health.
I know, definitely. But, a little bit bad for my study. But there's every time I go to Fresno to work, or, there's 2 or 3 patients with a potentially new diagnosis of syphilis. And again, are they recurrent syphilis? Is it, reinfection? Is it inadequately treated? Again, most of the patients that get syphilis are, have very complicated lives, right? And so either can't get treatment, can't get testing. You're not sure. Very few of them come in, with the classic like canker and all that stuff. So this study is to collect samples. That is my superpower, is collecting good samples and looking for new diagnostic markers of active syphilis and treated syphilis, and then hopefully congenital syphilis as well, where, I think the way we diagnose congenital syphilis is also based on it's like worse than the mom, I think, because you're waiting for the mom's RPR to to wash out, right? And so to look for new biomarkers that we can diagnose, get to a faster diagnosis at the time of delivery.
To expand on your superpower, like, one of the other grants that you have is a K-24, like, how to build a translational research career, so, K-24s, are really important for, like, mentoring the next generation, and I was just curious, what are the biggest barriers you see for early career physician scientists that are trying to bridge between basic science and clinical or translational research, and what advice would you give to them?
I mean, I think, I'm so blessed to have that K-24. I think it's, and I one of, I think the biggest resources I have in my lab for training is that we do have this huge biorepository. So I was talking a little, like, I think we have like 2,700 placentas from Uganda from this clinical trial we just finished. I have 800 COVID positive, like COVID infection placentas. I have another 300 vaccine exposed, placentas. So, with such a large biorepository, we can really answer specific questions and have the ability to select samples. Like whenever we run a big experiment, I think my lab, they might get sick of me, but we spend like 2 to 3 months on just sample selection. I just need to look at it multiple times. Like, did we think about this? Did we think about that? Because it's such, these experiments, these molecular experiments are so expensive, right? And they're so involved. And if there's too much clinical noise or the sample, we didn't think about some potential confounder, like, they have an autoimmune disease or, all this stuff that now we like go through systematically, then you don't know what to make of the data, and the data is very noisy, and your conclusions, I think, can be skewed. And so, I think that, and with this K-24, it was just awarded, so just a few months, but I, I the resources really were, to protect my time so that I have more time to focus on mentorship, and then really I think train future physician scientists to, what to think about with sample collection. You know, bad samples, good samples will all give you bad data. Everything will give you data. It's just whether or not the data is usable and whether it'll lead you to the conclusions that you want to arrive at, right? Because I think
Fantastic.
you want to
It's
answer
so
questions well.
Yeah, no, it's fantastic. Your mentees are lucky to have you for sure.
Yeah, I was
It's
thinking
fun.
that too.
It's really fun. I know there's new cohorts all the time. I have a Stephanie Cho's a PICU fellow, a pediatric ICU fellow who's interested in placentas and pulmonary hypertension. So we're looking at our placental data, and there's always like new things. And this is again where you just don't know where, we're Googling random things together like meconium and, and all this stuff. So I think that's the fun part of science. You just don't know where you're gonna go.
Yeah. Well, we've really enjoyed talking with you and learning more about, a wide spectrum of infections. I feel like I've traveled around the world in the past 40 minutes. We like to close on something fun about our guest research team. Is there a favorite music that you all listen to while you're looking through your placental samples? Is there a GA lab tradition that you would like to share with us?
Oh gosh, we don't it's pretty quiet lab, nobody listens to music. Um, People have their headphones, but there's no lab. There's no
It's
lab.
different, right? When I
Yeah,
was doing my PhD, we had, like, the lab CD player,
yeah.
and we'd go, like, through the options and we'd be like, "Do we wanna listen to Spanish guitar or Modest Mouse?" Or, it's like 'cause, of course, like, the laboratory is always I mean, one of the really fun things is it's a very international environment often, right? And so you'd
Yeah.
be like, "Are we gonna go to Japan or Argentina or New York today?"
I feel like a lot of the labs I walk through now are very quiet, everyone's doing their
It's
own thing.
true.
And
Yeah.
also the open lab space is just echoes. Right, the design's different. I think that's how we try to do something fun maybe 2 or 3 times a year, and but my favorites are we usually do some sort of hike. We do a Land's End hike or a picnic, and people bring their kids, and that is fun to see, people's families, and I don't know. It's kind of funny when you're around enough, but I think this is my 8th year as faculty, and I'm like, "Oh, maybe we need to do like a lab family tree," 'cause, at the beginning, it's just 1 or 2 people, and all of a sudden, there's so many people that then graduate from your lab, and we invite, a lab alumni, and sometimes they come and bring their kids that are bigger, so um, it's always fun to see the growth.
We had a Center for Fetal Maternal Medicine, which is Tippy McKenzie and Mary Norton's, efforts to build a precision medicine center for fetal maternal medicine, and there was the 10-year anniversary last Friday, and a lot of folks from my lab brought their, like, significant others. I brought my kids or dogs. It was really fun. There was just, like, this beautiful, like, display of, like, friends, families, and animals that people bought. So, I was thinking we
Yeah.
should do more of that.
No, it's fun. It's good. You're so immersed in the lab, and lab is kind of like a family, but you only see
Mhm.
like one part of it.
Yeah, that's true.
Um,
Well, we're so grateful, Stephanie, to have you on the podcast. And clearly, you're a leader in maternal fetal medicine and your translational research all around the world, local and like Betsy said, all around the world is really shaping how we understand immune development and placental biology and perinatal infections. So we're really grateful for you coming on and sharing your expertise with us today.
oh, thanks so much for the invitation.
Yeah, that's great.
All right. Well, thank you all for joining us, for the listeners too. And we'll catch you next time, our At the Bunch segment of the Incubator podcast. Take care.